Recording in
Good evening, Maggie. Can you hear? It's on. Good evening. Welcome to the Shoals Marine Laboratories Weekly Marine Science Seminar. I am Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and we thank you for joining us this evening for our 8th Rock Talk of the 2024 season. Our seminars are affectionately referred to as Rock Talk, in part because back in the early days of the lab, seminars and classroom lectures took place out on the rock. Today, we've moved inside, which is particularly nice for those of you in Zoom world. We are going to be experiencing a thunderstorm at some point, and so just bear with us. So we're now indoors, and that gives us an opportunity for our island community to come together and to explore interesting topics about marine science and our environment. For those of you joining us online who may be new to our lab community, the Shoals Marine Lab is the oldest and largest marine lab in the country. It is focused on undergraduate training, both in coursework and in research. The lab is operated jointly by the University of New Hampshire and Cornell University. In addition to our guests joining us remotely tonight, we have two courses on Appledore: Investigative Marine Biology Laboratory and Sharks Biology and Conservation. Do I need to go back into the original one? Okay, guys, on the first group, the first class again was Investigative Marine Biology Laboratory. We also have our summer undergraduate research group, affectionately known as the Surges, who are involved in summer-long research projects. We're excited to have everyone here, both in person and online, joining us for this seminar this evening. The format of the seminar will be a 45-minute talk, followed by 15 minutes of questions and answers. For our friends online, please use the Q&A box in the bottom of your Zoom screen. We'll read your online questions out loud for the entire audience. For those of you in the comments, we'll pass around this mic for your questions so that everyone can hear your questions. This evening's speaker is Dr. Doug Fudge, who is a professor in the Schmid College of Science and Technology at Chapman University. He completed a bachelor's degree in biological sciences and a master's in science and math education at Cornell University. Doug then completed a master's in comparative animal physiology at the University of Guelph in Ontario, Canada, and a PhD in comparative biomechanics at the University of British Columbia in Vancouver, Canada. He was a professor of biology at the University of Guelph from 2005 to 2016, and he is currently a professor at Chapman University in Southern California. Research in his lab focuses on the organismal biology of hagfishes. The work is multidisciplinary and integrative, employing concepts and methods from biomechanics, physiology, cell biology, proteomics, material science, and engineering. Although the lab's main focus is fundamental, discovery-based research, some of their findings have inspired practical applications, and biomimetic applications of hagfish defense line are now a major source of funding and inquiry in this lab. His interdisciplinary approach is evident in the range of journals in which he has published: Nature Communications, ASC, Biomaterial Science and Engineering, Journal of Experimental Biology, Evolutionary Ecology, Fish Physiology and Biochemistry, and Ichthyology and Herpetology. Doug took an undergraduate class at the Shoals Marine Lab in 1991, which set him on a course in marine biology that has involved research on animals such as bluefin tuna, baleen whales, and hagfishes. He spent 2 summers on Appledore working as a lab preparator, with one of his jobs being the preparation of the minke whale skeleton that hangs above us in the commons. That is thanks to Doug, so everybody should clap right now. He has taught at Shoals either as a TA or instructor for most of the summer since 1994. For the last 4 years, he has also served as the chair of the Shoals Marine Lab's Alumni and Friends Association Recruitment Committee, helping us to increase the diversity of students, faculty, and staff at Shoals. It is my pleasure to introduce my colleague and friend, and Gemma's dad, Dr. Doug Fudge, who will present his talk, Weird and Wonderful Hagfishes. Please welcome me in joining Dr. Doug Fudge.
Right. Thank you, Sarah. This is very exciting for me to be giving a rock talk, telling you about what I do, the research that I've done with my students about this. So I want to start with everything that about the work. Okay, start with a few acknowledgments so I don't have to rush at the end. So most of the work I'm going to tell you about took place at UC in Vancouver, University of Guelph in Ontario, happy to be where I am now, and the goal thing. So you're going to see goals fingers all over this research. I want to also acknowledge funding from NSERC, NSF, and DARPA, rank of the military. That's what I'm going to acknowledge all the people who did the work by showing a picture of them on the relevant slides. Try not to take too much time and telling you about you'll see it in the picture. Before I get into the biology, I want to acknowledge some really important mentors who were all. So when I took the course in 91, as Sarah said, Denny Taylor, who's not here now, but he's coming in the second week of the Yale. He was my professor, and Sarah Gould, the other instructor, they were just learned so much from them about how to teach and also how to do this. Two other people I have to mention are Rick Martini and Dave Meyer. These guys were both doing active research when I was here, and I think I was a lab operator. That obviously is what I'm going to hear more about the inspiration from them. At TJV, there was active slime. And hopefully you recognize that name. Iser is in that for him. Our second director. I'm going to start with just some basic facts. These are not things that I figured out, but just to give you a little bit of background. And then I'm going to launch into some rapid fire slides about the things that we figured out over the years. Okay, so hagfish basics. What is a hagfish from an evolutionary standpoint? Hagfishes are jawless fishes. And what that means is that they belong to a group that arose over 500 million years ago. And they gave rise to the jawed fishes, which is what we normally think of as fishes. But there was a time when the jawless fishes dominated the oceans on this planet. Only two groups of jawless fishes are still around. These are the hagfish. Most of you know this now. Hagfishes are scavengers. When something dead falls in the bottom of the ocean, it's usually a hagfish in their part. Sometimes they're also predators. What eats hagfish? Several important groups rely on hagfish. We've seen a list here: toothed whales, sea lions, seabirds, and octopus. This video on the right display its own. Hagfish minding its own business, and well, comes a female elephant seal that just sort of clears it up. One thing you might notice from this list of hagfish predators is that there are no fish. I'm going to tell you why that is. When talking about hagfish predators, we have to talk about humans because humans are making a big dent in hagfish populations around the world. It's mostly Koreans who are eating hagfish and making leather. There's fisheries all over the world shipping hagfishes to Korea. This is outside a hagfish restaurant. Okay, another cool thing about hagfish is they tie themselves in knots. It's an unusual thing for any organism to do. This is one of the great planet secrets of all time. It was mid 80s, I believe, probably 86. And it was so popular that they brought it out as like a digital gold speaker they could buy. So obviously, hagfish don't do all these. Bit of a joke. My favorite is the square knot, which involves two hagfish. That would never happen. But they do make two different types of knots. We didn't know when the first hagfish are not a knot. We knew that they made overhand knots, but more recently, this paper, Amy et al. 2019, showed that hagfish can do two kinds of knots: overhand knot and a figure knot. Use those for different knots. So the knotting is used for clearing slime in their bodies if they get trapped in slime. For gaining their stuff. We heard about carcass, and if you've ever tried to hold on to it, you know that they very easily get out of your grasp, like knotting. Okay, most of you know that hagfish are known for their slime. This picture gives you an indication of ridiculous hagfish slime. They can make huge volumes of slime in very small amount of time. We'll talk more about that. The best way to introduce you to this phenomenon that hagfish is sliming is to show you this. Show you this video. This was taken by some colleagues off the coast of New Zealand, about 700 meters down. We'll look down at the data camera and watch the sea. It's mostly a biodiversity study. And what you're seeing here is New Zealand hagfish being chomped on by a sort of related to dolphin. Actually, more related to sharks. Kite shark. Okay. I'm going to play that one more time. But watch it again. So watch what happens when the shark bites the hagfish. There's this cloud of stuff that appears. That's the slime. You'll see it again in slow motion. It floats out. Okay, so see all that cloudy stuff now clogging the mouth. This was not a good decision by that shark. And obviously it was totally unsuccessful. So I'm going to take probably the next hour about slime. And there's a lot to unpack. I'll actually be skipping through. I'm not telling you all the stories. But we need probably a full half hour to tell you. The way I'm going to structure this is posing questions. And the reason I'm doing this is mostly for the IMBL class, because we just started talking about scientific methods and the importance of questions. So I structured it around questions so that see the and hopefully get some answers. So here we go. A lot of them are going to just be really simple, no-brainer questions. Some of them are going to be really tough. So what is it made of? If you stimulate a hagfish slime gland electrically, cause the muscle around it to contract and flip. Take a little bit of that slime gland exudate, and put it on the microscope, you'll see something that seems sort of football-shaped structure called a skin. They contain a coiled protein fiber that raffles when the slime interacts with that product you saw in the parks. And there's a close-up of one of those stains on the right. There's also mucus about equal quantities as threads, and that's what that sort of granular stuff is. Okay, where is it produced? I already alluded to slime glands. This is a picture of a newly discovered albino hagfish. I'll tell you more about it. It's called the ghost hagfish, or it's fantastic. And it's an albino, and what that means is the young hagfish are pretty transparent. So what you're seeing here is a young ghost hagfish. And the reason I'm including it is because you can very clearly see those pillows of white, those are slime. And the data below show that the slime lines are much bigger near the head and the tail, quite a bit smaller. Let's zoom in now on the slime glands. So this is the histological preparation of the section. You're seeing two slime glands noted by the arrows there. So pretty big structure. 3% of the body of the head. And there's even more of a close-up of a single slime gland. If you really zoom in, you'll notice that there's two types of cells within that slime gland. There are mucus cells, which are pale, and then the red ones are the red cells, which are red cells. Okay, one of the questions that I tried to answer during my PhD was how much could they actually make if they let it all fly all at once? And the answer is 24 liters of slime, right? So a liter, a kilogram of water. 24 liters for a hagfish that only weighs 60 grams. Okay, so they're making a total amount of slime that is 400 times their body. Okay, question. Don't ever do this all at once. Okay, I'll show you some video that shows that they only really take slime that they're getting. And so they would never just let all their slime go at once. So then the question becomes, okay, they make so much of it, how would that even possible? Something they're times larger than your own body volume. And this is a bunch of hagfish fishing boat with plenty of water to make slime. One of the things I figured out during my PhD was that hagfish slime is incredibly dilute. If you measure the amount of threads, silk-like fibers that are in there, and you measure the mucus, it's about 20 milligrams of each of those things in a liter. Okay? So like 40 to 50 milligrams of stuff in a liter of slime. So if they made their slime as concentrated as like make your salivary mucus, they would have to make 1,000 times to get this contribution by the mucus and threads. Okay, so what about the structure? It looks, those of you who took the slime on the boat today, it's not really clear what's going on. There's fibers running through, there's mucus everywhere. How are they doing this? How do they make something that expands so much and so quickly? So we did some work with the kind of monitoring in her postdoc, Daniels. And they have this really cool laser scanning setup that they use for looking at the structure of animals like jellyfish. And so we had a hagfish in an acrylic box like this. We did a gentle pinch on the tail and it left a little massive slime in the box and closed it up and scanned it with their laser system. And this is what we saw. This is not public, we can clean this up, but we were shocked to see how aligned fibers are. That is not something we ever expected to see. And that's opening up many ways of thinking about how this slime is built, how it deploys so quickly. They actually set it up with about time it comes out of the gland. We're also trying to look at the ultrastructure of the slime. They're really high power. So this is a false colored microscope image, scanning electron microscope. Colors denote the threads, and then okay, so the threads are green. And these images are sort of changing the way we think about how the slime We don't know if there's a lot of preparation that went into getting these samples. We don't know if this is the true structure of the slime or just an artifact, but it is pretty cool looking. Okay, what is the function of the slime? This was a question that we tried to tackle with very talented undergrad when I was at UBC. She was undergrad. She did these experiments on fish heads and exposed fish heads to hagfish slime and measured the resistance across the gills. It showed that the slime is really good at clogging gills, slowing the flow of water, which was our main hypothesis. That was Jeanette's work. This work made its way into a book that you probably all have a copy of at home. The Secret of the Suffocating Slime Trap. It's basically about gill clogging by hagfish. And there's a cool picture of holding the slime there. And you might recognize that person in the foreground. That's Gemma holding the slime. So it's perfectly on the cover. Don't worry, that's the last one. I will embarrass you. Okay, I'm going to tell you a little bit more about the fun. Fish head experiments were really hard to do. And we had lots more questions about what the slime actually does, how it actually evolves. So we designed this really simple apparatus with a net on the bottom, hanging from a load cell. We add the sample and measure how fast it flows through that vent. If it's good at clogging, it would be very slow. If it was crappy at clogging, it would run. Okay. So a lot of people worked on this, and we collected some pretty cool data. The lines on the left are for slime samples. And what you're seeing on the x-axis is the total concentration of the stick. And then on the y-axis is the drain rate. It's indicative of how well it's clogging. The low drain is clogging. You can see that hagfish slime is clogging really well at values between 10 and 100 milligrams per liter. We compared this performance to other materials that you might think would be good in clogging, like high molecular weight polymers, food thickeners, things like that. And those are the three curves on the right. And we need 100 to 1,000 times more of those materials. So somehow I figured out that clog gills very little. We also wanted to know, what's the mechanism? It seems like they're doing something really special here. And when we thought about it, we came up with what we thought were reasonable hypotheses. The first one is called the obstruction hypothesis. We also call this the shower drain hypothesis. Basically, what it proposes is that the threads of the slime feed the holes in our mesh. That's how they slow down the water. Another hypothesis is that the mucus viscosifies the water and just makes it slow down so it so and then the third one is called the soft elastic membrane hypothesis. This one says that no, it's not about interacting. So three distinct hypotheses, and fortunately, they make distinct predictions. Okay, well, at least two of them make the same prediction. The first two predict that if you make the holes bigger in your mesh, then the drain rate should go up. Or if it's like a bath drain, be less good at the bottom of it. The last one predicts that whole time, as long as the threads are long enough to stand, it should still work. The resistance of the slime. Okay. So we did a very simple experiment. We measured the drain rate for hectare time over a range of hole sizes. And what we found is that it's almost insensitive block really small holes and block really big holes. Performance doesn't improve. That's consistent with that third hypothesis, and inconsistent with the first. Hopefully, I am able to pull off this term. Okay, more about clogging. So we also wanted to know which part of the slime is doing the clogging. Mucus, is it the threads, put two of them together, and we were shocked to find out that the mucus is doing all of the clogging. I had a very thread-centric view of that, and this got me really. Okay, so if the mucus is doing the heavy lifting, what the hell are the threads here for? Okay, so we answer one question and then the new one pops up. Okay, so what's the one thread? If we thought about it, we realized that it's probably to hold this line in place so it doesn't get blocked away. We did some experimentation with this. Added the slime and then added seawater after it to see how well it did with multiple flushes of water. If you leave the threads out, it'll clog pretty well the first time, but with subsequent flushing, it just goes away. It's like seawater. So yeah, so we think that the threads aren't involved in clogging per se, they're involved in making the slime on the gills where it appears to be. Okay, these gland thread cells are pretty amazing. All of them related to the stain. For a long time, we stare at these SEM images of games and wonder how are they what's going on there and how does the cell actually build it. So it took us a while to figure this out, but we used a technique called Fudge. And when you do this, you're basically milling through a sample. And then you're taking a slice and then taking an image. Slice it. By the time you're done, you've vaporized your entire sample. You've got this nice image stack. And from that image stack, you can sort of look at things in 3 dimensions. So here we are milling through one of these cells that makes an active fiber. And I don't know about you, but I'm staring at this for honestly still. So what we had to do was to follow one piece of the thread as far as we could. And it turns out following one piece of thread so gave us the error. We started with one structure. So one of these loops is highlighted in green here. So there's about a dozen of these described in this video. A fully mature hagfish skate is about 5 minutes. Okay, so that was nice. We figured out how it's actually put together. We still don't know the mechanism whereby the cell and that's another question. We have a hypothesis for it that involves a spinning wheel-like mechanism that involves a cell nucleus. It's a totally untested hypothesis at this stage, but it's the best we can do at this moment. Okay, I mentioned before that the slime gets ejected out of the slime lens and it sets up 400 millisecond lens. What is that? So let's watch some videos from, this is a hagfish that we've gently placed back so that we could clearly see the slime lens going on, stimulating with the there. Jets of exudate shooting out. We're not capturing the full event, the hydration where it swells by 10,000, but that's gives you an indication of what happens when they get attacked. They shoot it out using muscular contractions. This is a question that people asked me for many, many years, and I didn't have an answer. And then I had a PhD student who tackled this question, and it's a very simple answer. The answer is it takes about 4 weeks for a hagfish to fill a slime gland. Fortunately for them, they have lots of glands. They don't eject everything every time. So they have lots of reserve capacity. Another thing that is super interesting to us is how do you unravel one of these structures at the bottom of the microns long? It could be 150 millimeters long, 1,000 times longer in 400 miles. So we started by looking at what these things do on the microscope. It's seeing this slime there. That's my microscope. But we know that the slime sets up in 400 milliseconds. This is taking longer. So what we think is even more important than this sort of spontaneous unraveling is a sort of peeling out of the thread, external forces as the slime is set up in seawater. So we did these very delicate experiments using single skeins and glass rods. We monitored the bending of the glass rod to measure how much force is required to pull the thread out of the skein. And I know you're dying to know what the answer is. The peeling force is about that seems like something. It's actually important for us to model the biophysics of how this thing works. Okay, we've also done some things with microscopes hooked up to high-speed video cameras to try to see the deployment of the slime in action. So this is video taken, I think it's 1,000 frames per second. It's going to look like it's happening slowly, but it's actually slow down. What you're seeing here is the mucus and the milky white stuff, and then those football-shaped things are the skeins. And these are giving us our first glimpses into what's happening in that 400 milliseconds. Okay, what about the mucus part of the slime? It's packaged in these disc-shaped vessels for about 5 or 6 microns in diameter. They're packed with this mucus like glycoprotein. When they get exposed to seawater, high-speed video under the microscope, swell up really quickly and it gets slow. But if you actually trace the swelling of those vesicles, you see they're down 200 milliseconds. So that's fast enough for 400 milliseconds. Another question that we just recently took a stab at is how did the slime evolve? Where did it? And really the only reasonable hypothesis is that it came from skin. That rarely happens. The reason we thought the skin is important is that there are 2 types of cells in hagfish skin that are suspiciously like the slime. There's a thread-producing cell, and there's a large mucus secreting cell. And you're seeing those here, ETCs and LMCs in the blood. And David Pertetsky, who I met because he gave a rock talk here several years ago, we ended up writing an ancestry together and doing a lot of work together. And here are some data that he generated in his lab. Caleb, some of you met here for one day, and actually with us. Dave's grad student. And these are transcriptomic data that look at the genes that are being expressed in the slime gland and in the skin. And you can see down below are the genes that are popping up in the slime gland analysis, and the top are the ones in the skin. And if you look for protein, you know the identity of the there's just a small number of them expressed in the skin, and there's a huge number of them expressed in the slime. So we think that represented, or that's indicative of duplication and diversity of red genes in the skin, giving rise to this amazing structure. A very recent genomics paper on hagfish just came out, and they found a very similar result. They found lots of red expression in the slime gland, not so much in the skin. Okay, so now you know that the epidermis has these two secretory-like cells, red cells and mucus cells. But what the heck are they doing there? Why do they need reds in their skin? So our hypothesis is that when hagfish get bitten by predators, their skin gets thin. And when that happens, that's when we think that the epidermal mucus is formed by the rupturing of damage of the skin. And so what you're seeing on the left there is a needle that goes straight across the skin. When you do that, you end up with epidermal secretion that is not hagfish slime. No, it's a different slime, which is the hagfish. Right. All right, I'm gonna go like that. I gotta show this. This is one of these epidermal red cells. It's showing you sort of glomerule-like red that's in the cytoplasmic cells. In the middle of these cells, we never noticed this before, but there's these granules that we don't know what they're doing. So that's the next thing, which is what is the function of the granule? Okay, I mentioned that we've gotten some funding from DARPA recently to explore the possibility of making synthetic slime. We've been working with engineers at the University of Illinois. I call them my partners on slime. And they are doing an incredible thing with making synthetic here of building a skin that can travel, sort of like. Okay, so that's it for the slime. I'll try to do the rest. So you might have noticed from that video that the slime was kind of amazing, but maybe even more amazing is that the hagfish doesn't die for being on by height and sharp. That was confusing to us. Super interesting. So we hypothesized that hagfish skin is super puncture resistant. So we actually tested that idea. Here, hagfish skin to other fish and found that it's not acceptable great immunity. Okay. So we had to scratch our heads and think of another hypothesis. And knowing a little bit about the body structure of hagfishes, we started to think about their weird relationship between the skin and the body. So you might have noticed this, those of you who saw hagfish today. Hagfish skin is almost like loose pajamas. It's not well attached to the rest of the body. And so the hypothesis is that there's plenty of room for the body to sort of get out of the way if a hagfish is attacked by a shark. And so we built this looking device on the right to drive shark teeth into the dead hagfish. We actually pulled the trap out here. It's been left out too long because weather prevented us from getting it, and there were a bunch of dead hagfish. So that's how we did these experiments. And this was the work of Sarah Fudge, who was a high school teacher. He took 2 years off to do a master's program with me. And what she found is that, yes, if you drive a shark tooth into a hagfish, it will go through the skin, but it will not damage anything else. It just slips in between the skin. Okay, this one, how do hagfishes multi-type face? This is a video taken on the Heiser. And you saw a version of this today. The reason I'm showing it to you is if you look on the left-hand side at the end of the video, you're gonna see a hagfish poking through one of the tiny ventilation. There it is. Yeah. Okay. That was an observation that the students made that they were really interested in. And they were like, what is going, how did that hagfish possibly squeeze through such a tiny hole? And so we actually did an experiment on that scene and continued to do this research back at the University of Washington, students there. Basically, we created escape rooms for hagfish to see how small a hole they could escape from. And the video of a hagfish figuring out a way to get through this. I'm not going to show you the whole thing. It would be really satisfying to see it get out of here. So that's. And I put Andy Turco on here because he was teaching with me at the time and he got obsessed with this observation of the hagfish leaving through the ventilation. Okay, another shoals influence question. How do hagfishes burrow sediment? And this was inspired by a video that Rick Martini and John Heiser showed me a long time ago. This was taken in the Gulf of Maine from a submersible Johnson Sea Lake. Now, I hear this sound.
See, he gets his head in, then he thrashes real hard. And if he's going to burrow here,
So he's
pretty
gonna
soon,
go under that rock.
yeah, pretty soon his body will stop thrashing and he'll just suck in. So something's happening with the anterior part of his body once he gets it in there. Great shots though. Yeah, because on the 3/4, this is. Now, look, goes just about. Before he's gone, it would be nice to get an 85.
Yeah. Well,
I don't ask much, do I? See how fast he's going in? It's like someone sucking up spaghetti.
Yeah.
Let's just see what.
Okay, so that's amazing for me to hear JB's voice in this room because he had such an incredible booming voice. And you know, we listen a lot. We made some progress on this project by getting hagfish to burrow into gelatin, okay, unflavored gelatin. And this has been used to study worm burrowing in the past. And amazingly, we got this to work out here with IMBL a few years ago. So what you're seeing here is a hagfish burrowing into gelatin. It's transparent, so we can actually see what's going on, unlike the video that you just saw. And we just had a paper published on this. So I'll show you a couple videos of this. This is sort of a typical burrowing event. And this is sped up 15 times, so we don't have to do the whole thing. But it looks pretty similar to what you just saw narrated by David Heiser. There was a lot of thrashing at the beginning, and then the thrashing stops, but it continues to make progress. So it's doing really interesting things within the gelatin. And again, we don't have to play the whole thing, but it does eventually make a U-shaped burrow and get to the other side. So we looked at about a dozen of these and sort of traced them so we could see, you know, the sort of average shape of the burrow. They're sort of U-shaped. Average burrow time was 7.5 minutes about. Here's some more close-up videos. This one's sped up 4 times. We're seeing a lot more detail about how that hagfish is moving through the thing. We also use something called photoelasticity, which allows us to see where the hagfish is pushing and how hard and when. And that really helped us understand the mechanism of the video of that. So this technique uses cross-polarizing to create a dark field effect. And wherever the gel is deformed, it shows up as a bright spot. You can see like where the gel is being broken up, where they're moving on the part. Really critical. Okay, I'm going to go really fast now. Tell you a little bit about hagfish reproduction, but really the take-home message is nobody knows about hagfish reproduction. Okay, Rick Martini is really interested in it, still is, and we just don't understand what's going on. So here's a video from the Tokyo Sea Life Park. I think it's the only video ever of hagfish hatching within an egg. And if you thought adult hagfish were ridiculous, this is ridiculous looking animal. When I look at this, I think they must be hatching in burrows because that thing would not survive for a second. On the mud, a lot of them do. So I think that's really interesting. If we're going to understand reproduction, yeah, understand burrowing, as we just saw. We have some new insights into how to study them while they're burrowing. This image on the left is a paper from 1899. For a long time, it was the best paper on hagfish reproduction. It actually had fertilized hagfish eggs, and these are drawings of those specimens. It wasn't until a century, more than a century later, that anyone was able to study fertilized hagfish eggs. This is a paper published by Martini and Pizer and Mike Lester on demographics of hagfish populations out here. One of the puzzling things about their data is that they got 10 times more females than males. That's weird. We don't know why that is. The eggs are super interesting. They have these little Velcro things sticking off each end, which are really good for allowing them to hook onto other hagfish eggs. But they also can cover them in this jelly-like mucus, which prevents them from attacking other eggs. This is super interesting to us. We have no idea why they want such exquisite control over how well the eggs attach. So stay tuned for that. We're looking at the biomechanics of attachment in these structures. This is a close-up of a few of those little Velcro hooks under the microscope. And you can sort of see how they interact. One of them slides along the groove in the other one. And then they have the lobes that sort of hinge. We're now making 3D printed hagfish eggs so we can study sort of their biomechanical properties. And out of time, but I will tell you for a few minutes about the hagfish biodiversity. So there's 90 species of hagfish, and that number goes up every year by 1 or 2. You can see, we're in a golden age of hagfish discovery. This is the cumulative number of hagfish in the world, and it is on a steep rise. We went to the Galapagos in 2019 to collect slime from 4 different species of hagfish for an NSF-funded project on slime diversity. And there were 4 described species of hagfish. And okay, so here's some pretty pictures of Galapagos, the team that collected all the hagfish, did all the analysis. And these are the 4 that we were targeting when we went there. The amazing thing that happened was we caught 4 other species that had never been seen before. Okay, so now there's 3 genera, 8 species of hagfish in the Galapagos. It's a ridiculous number of hagfish in a small area. We don't know why. This should look familiar to those of you who went hagfishing today, but this is the same process in the Galapagos. Opening a hagfish trap that goes down probably around 700 meters, so 7 times deeper than today. And if you look carefully, there's several species of hagfish in that one trap. I mentioned the ghost hagfish before. It's in Tasma. It's amazing hagfish. This is a video, the first ever video of ghost hagfish. One in the background is another species that is not albino. They're not both relative, but they always seem to be together. That is. One of the hagfishes that we discovered, we named it after my PhD supervisor, John Gosling, where I did my PhD on the line. And here's epicleated dog line eye. And then I include this for the sharks class because we have a little photo bomber here. Of course, the hagfish are way more interesting. And you're going to see in a second that shark from the back. And I want an ID from the shark class. See this? Okay, that's not a shark. That's another photo bomber. Anyone? Anyone? Yeah, what's the common name? Yeah, broad nose. I think it's a 7-gill. A 7-gill. Yeah. And then I'm going to end with this slide. This is another species that we discovered and described, and we named it martinii after Rick Martini, who's one of the people who introduced me to hagfishes way back when. So I will stop there. I've gone over time, but hopefully we've still time for questions.
We do have time for questions. Jasmine, waiting for like a thing.
Another one?
Did you test the unraveled rattles in freshwater vessels?
In saltwater, because we're interested in how it works naturally. They will unravel in freshwater. But it's sort of more explosive because the aquatic gradients are higher. If you add hagfish slime exudate to freshwater, it does make something like the natural stuff, but it's not quite as good.
You mentioned that the hagfish are being eaten and used for leather in Korea.
Yeah.
That seems strange to me, given everything that you said about them. Are they meaty? Like, what is special about their skin that it's like being
So
eaten
for
and used for
food,
leather?
my impression about the food thing is that there's hagfish restaurants, and they cater specifically to couples who are having intimacy trouble. And so it's like this phallic thing, yes. And so it's going to revive the marriage if you go to hagfish. That's what I've heard. I don't know how much of that is true. And then the leather, they make these strips. So you might have seen wallets that say eel skin leather. It's actually hagfish.
The hagfish like intentionally slime their attack, or is it an automatic response?
It's a really good question. Could everyone hear that? Are they intentionally? Question was, are they intentionally sliming, or is it just a reflex? The reason I find that so interesting is because during those hagfish Houdini trials we did, they did not slime, and we saw how squeezing themselves like through really tight spaces. So if it was just a kind of mechanical trigger, you would think the slime glands would go off. So that implies to me that they have some kind of central control over when they slime. Great question.
Do they ever like eat it themselves and accidentally like hit on their own slime?
Not choke on it, but they can get trapped in it. And that's when they tie themselves in knots that wipes the slime off.
So you mentioned that with the sort of clogging research and trying to see like how good it is at sort of, clogging pores and things, that you removed the fibers and were testing just the mucus.
Yeah.
How did you do that?
So we can collect the exudate into a special buffer. We call it stabilization buffer. It has high salt content and secrete planets. And that keeps everything from exploding, keeps everything in that state. We can then filter that stabilized slime. The vesicles go right through, and then the threads are retained on the filter. So we can make a preparation with just the meetings.
You mentioned that you got some more insight into the structure of the skeins. Do you have any more insight on how they release the thread without like tying themselves with knots?
How the threads themselves deploy? Yeah, that's a really good question. So the way that it's built is about as good as you can get in terms of knotting. So if you talk to anyone who works on boats, there's this thing called flaking a line or a chain where you put it down in a way that you can pull it out. It won't hang. Basically, I figured that out.
Okay, okay, cool. Hi, I'm Rowan. I'm a surge here. Thanks so much for the talk. I was curious if you about the evolutionary history. You kind of just touched on it a second ago, but like, what are I came in slightly late, so I may have missed it, but what do you think are the evolutionary factors that cause hagfish to evolve the way they do? And do you think they diversified at that time, or that they both diversified relatively recently in evolutionary time scale?
Great question. So the answer is both. So there are people who are working on evolutionary history, looking like the genus Mixini, after treatise, two large genera, that they split maybe over 3 million years ago. And yet, the ones that we caught in the Galapagos, there were 4 of them that are closely related to pairs. So we think their common ancestor for colonized Galapagos then split. Not very long ago. And in terms of like what were the changes that accompanied those transitions, I think adopting a burrowing lifestyle was a huge thing, especially for things. It's not all the applicants or that the question was about impetus for burrowing. Yeah, I mean, if you look at the kinds of things, Karen knows a lot more about this than I do. The kinds of animals that have a burrowing lifestyle and most different, but they're about that big. Well, occasionally you get stuff. But hagfish are kind of huge. They have amazing hypoxia tolerance. They can go something like 36 hours without oxygen. So that allows them to burrow into a dead whale with 0 oxygen. Hang out in a burrow with very little oxygen. Okay, was there another one back there?
Yeah. So you said that they have predators like sea.
Yeah.
Oh, yeah. How does the mucus not really affect them? I mean, like they don't have gills to clog, but I would imagine it's still not nice to have mucus all up in your mouth.
Yeah, so the way the slime works is like if you're a fish, the fish structure is pretty strange if you think about it. When you're eating something as a fish, whatever you eat goes right past your gills, right? So if you have a clever prey like a hagfish, that's an opportunity for them to maybe not be eaten. If a sea lion comes along, they can slurp you up. There's no gills because they breathe, they breathe air alone. So there's no risk of the slime clogging it. But they just slurp them up. And it's probably pretty slimy in the stomach, but eventually they can digest them.
Hello. Hello. Okay. I was curious about the fact that they knot themselves. What does it look like when they unknot themselves? Because for something that doesn't have arms and hands, right? How do they do it? I don't know if they're just so lanky that they like happens on its own, or how does that work?
It's pretty amazing. So if they're covered in slime, they'll start at one end, usually the tail. And they'll create the knot and then they'll pass the knot. They'll pass their body through the knot. And so when they're done with that, they're not knotted anymore. And the slime is wiped away.
So we're out of time for questions, but I'm gonna ask one more. How many hagfish did you get in your hag fishing?
IMBL, how many hagfish you get? 28.
Excellent. For those of you in the room that did not already hear this, I have never seen a live hagfish. Tomorrow is my day, and so I'm super, super excited. I really want to thank Doug for giving our rock talk this evening. I think it was amazing to hear more about a group of fish that many people have never heard of, much less had one named after one of the faculty members here on the island. And so many different questions. I'm hoping that the students in the room are seeing that questions always lead to more questions, and that you can spend an entire career studying things that just keep coming out of the work that you've been doing. I think we demonstrated that beautifully tonight, Doug. Next week is on the website. It says that our rock talker is TBA. I will announce right now, our rock talker will be Dudley Dudley, who was a New Hampshire state representative at the time that Aristotle Onassis was trying to put a deep water oil unloading station here at the Shoals and then a refinery at Durham Point. This is the 50th anniversary of when that happened. That was in 1974. And she has agreed to come out and do our rock talk next week. So for those of you online, that is who will be speaking next week. And for those in the room, that's who will be joining us next week. I want to thank everybody for joining us this evening.